Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be discussing something that's really interesting, and that is how do the billions and billions of cells communicate within our bodies? So we're going to be talking about cell-to-cell communication locally, systemically. We're also going to be looking at a concept called signal transduction. Once a cell receives a signal, what does it do with it? Thanks for being here. So the first thing we're going to talk about today is local cell communication. This is super important. There's a few different ways in which this can happen. The first way that this can happen locally is paracrine signaling, and this is when a cell sends out a signal. Again, it's local to other cells that will help change the behavior of those other cells. A really good example might be growth factors that tell other cells, hey, it's time to grow and divide. Another way that cells communicate, well, sometimes they communicate with themselves, and that is a process called autocrine signaling. This can be common in immune system cells and even cancer cells. Another really important way that cells can communicate locally is through direct contact. And this is really important for our immune systems, where one immune system cell can communicate whether or not they've found something that is bad inside of the body. It's also a way for our body cells to signal to the immune system whether or not they're healthy or sick. Perhaps the closest method of contact, the most direct method, is here, and that is when cells exchange communication signals through little tiny channels that are connecting the cells together, such as gap junctions or plasmodesmata in plant cells. The bottom line is, when we're talking about local signaling, we're talking about a cell that sends out a signal that is only impacting the cells directly around it. Now there's also long distance signaling, and a really great example would be the synaptic signaling from the nervous system. Here you can see a nice picture of that, where a nerve cell sends out a message to a receiving nerve cell, and that recipient is then going to fire that nerve impulse along the length of that cell, and it's going to in turn signal another cell to do the same thing. Another way in which this can happen is endocrine signaling. A lot of times we're looking at hormones here. A really good example would be when the pancreas sends out insulin after our blood sugar rises after a meal. That sends a signal to the pancreas, hey, we've got to get the blood sugar out of our blood and into cells. In response, the pancreas will then send out a signal, i.e. insulin, and that insulin will allow cells to bring in that sugar, thereby lowering the blood sugar levels. Now, if we were to compare and contrast the two then, when it comes to local versus systemic signaling, in terms of distance, obviously the systemic signaling is going to cover a lot more ground. The methods are going to be a little bit different. With the long distance signaling, you've got nerve impulses from the synaptic side of things, and you also have the endocrine system. But for local, you have paracrine and autocrine, and we also talked about the juxtacrine, where you've got the immune system cells that are communicating directly with one another. And of course, you have the gap junctions and plasmodesmata, which are direct cell communication networks that tie cells together. So let's take a few minutes and talk about what happens to a cell when it receives a signal. And that's the process of signal transduction. And there are three steps. There's the reception of the signal. We're going to talk about a couple of really important proteins that are involved in that, receptors. The next part is the signal transduction. The cell has to interpret and amplify that signal inside the cell. And finally, none of this is going to be very useful unless the cell responds. And that's the third part, cellular response. So let's take a closer look at each one of these things. So in the reception step of signal transduction, I'm going to focus on just a few really important receptors. There are more, but this will give you a pretty good idea of what cells can do. So the picture that I'm showing you right now is a G-coupled protein receptor. And the reason it's called that is this receptor is going to use an energy source called GTP instead of ATP to signal this receptor. And so you're going to see on the upper left-hand side, what we have is we have a receptor. That's the yellow protein that is embedded within the membrane, and it's going to attach to a ligand. The ligand is the thing that binds to that receptor, and it is a very specific binding. You're going to see Inside the cell, on the cytoplasmic side of that membrane, you've got GDP. Now, GDP, like ADP, is not going to do a whole lot. We need GTP in order to perform a reaction. And so once this ligand binds to the receptor, what's going to happen is the GDP is exchanged with GTP. That activates the receptor, and then you're going to see a couple of parts break off from that and go to this adenylyl cyclase. Once that happens, the adenylyl cyclase can then use ATP to start the cascade and begin the signal transduction step inside the cell. But the main point here is that when you're looking at a G-coupled protein receptor, it is a receptor, and it's using GTP in order to do the work initially. The second type of receptor that's also very common is known as an RTK, receptor tyrosine kinase. Now, this one's a little bit different. It's not using GTP anymore. We're back to ATP, that energy currency inside the cell that we're used to seeing. You're going to notice that this is a dimer. In other words, it's got two parts. And after that ligand binds to the RTK, as you're seeing in the upper left-hand side, those two parts come together. That's the dimerization. And then you're going to notice ATP is being utilized to initiate this receptor in regards to having it actually do something inside the cell. Once that dimer has been phosphorylated, it is going to power a reaction. And you can see on the right-hand side, you've got an inactive relay protein becoming active. And once again, this is going to start the next step, that's signal transduction. And eventually, we're going to get to a cellular response. Now, there are other types of membrane receptors. The one I'm really going to focus on here is the gated ion channel on the far right-hand side. I'm not so concerned about these other three, but that gated ion channel is, as the name implies, using ions, and those ions enter the cell, and that is going to start a signal transduction and a cascade. But you're going to notice here that these ions are brought in by the binding of a ligand on a cell surface receptor that opens up this channel that allows the ions in. So once inside the cell, the signal transduction pathway doesn't always necessarily look like this, but you can see here there's a lot of different steps. At the very top-hand side, on the upper left-hand side, we've talked about step one. That's the extracellular signal, that ligand binding to either a G-coupled protein receptor or an RTK as an example. And that's step two. We have the receptor that is on the cell membrane. Then we have step three, the intracellular signal activation. Now, the signal transduction pathway and signal transduction in general, usually when I use those terms, I'm starting with step four, and that can be the release of second messengers. Now, we've talked about the smooth ER in the past and how the smooth ER can store calcium. Well, calcium can be a secondary messenger. There are other molecules that can serve that purpose as well. But generally what happens is that activates a whole bunch of protein kinases, something we're going to talk about in a few minutes. And then eventually we get to transcription factors. In other words, at this point, by the time we get to steps 6 and 7, we're dealing with the response side of things. So what does the response look like? Again, all of this is irrelevant if the cell cannot respond to the signal that it's received. If somebody gives you information to do something and you don't do it, well, the information wasn't all that valuable. And that's the same thing that's happening here. And so in the response side, you've got here, let's say this is the nucleus of a cell. We've got some genetic information there, and the DNA eventually needs to be transcribed into something called RNA, which is a set of instructions to make proteins. The response side is usually going to activate some type of a transcription factor that is either going to turn a gene on or turn a gene off in response to the signal the cell has received. And so if we go back to our example of our blood sugar, let's say we have a snack and our blood sugar rises, what has to happen is the pancreatic cells need to receive a signal. They go through and they receive it, they transduce it, and then eventually what we have is a change in the expression of the genetic material. In this case, maybe it is the genes that are responsible for coding for insulin. Well, that transcription factor then turns on those genes, and insulin is going to be produced as a result. Once blood sugar is lowered, there's going to be another signal sent to those pancreatic cells that says, hey, you know what? We don't need this gene active anymore. And so there will be another signal received, signal transduction, but the response this second time will be different. That will be to shut the genes off so that no more insulin is made because blood sugar is returned back to normal levels. All right, now there are a couple of really important enzymes that I want to discuss in signal transduction. One is the protein kinase. We've already sort of discussed this when we talked about the RTK, the receptor tyrosine kinase. But what does a kinase do? It adds a phosphate group, usually from ATP, to some substrate. When that happens, it's going to change the behavior of that substrate. And so when we talk about metabolism in general, we often say that ATP is the energy currency inside the cell, and that ATP donates a phosphate group to something, usually a protein, possibly an enzyme, and that allows the enzyme to do work. And eventually, at the end of the electron transport chain, that phosphate is restored to ADP, restoring it to ATP. But we don't really discuss how the phosphates actually move around, and it's not a magic trick. What's happening here is in this particular picture, it's a very simplified picture, but it gets the point across, and that is you've got a substrate over here on the left-hand side, and it needs to do something. Let's call it an enzyme. So that enzyme needs to perform a reaction, but it can't do that until it is activated by a phosphate group. And so that protein kinase is actually the enzyme responsible for transferring the phosphate group from ATP to the substrate here, leaving us with ADP. Now that's all great, but what happens when we need to take that substrate and return it back to its original form? That means we need to remove the phosphate, and that is the job of the protein phosphatase. It's going to remove that phosphate. It will return that enzyme or that protein back to its original state so that another phosphate can attach and it can do its job again. All right, everyone, so that was a quick rundown of how cells receive signals. They can be local, they can be systemic, and then specifically what they do in the reception process, the signal transduction, and of course, the response. As always, thanks for being here and have a great week.